Hi, good morning. My name's Grant Burgess, and I'm delighted to be able to present to you today, although I'm sorry I can't be there in person. I'd very much like to thank Professor Mike Reed and the BOA for giving me an opportunity to present our work here today. And I'd like to begin by showing you a brief overview of what I'm going to talk about. So the key messages I want to really present to you today are that we're going to be talking about bacteria, of course, infectious bacteria. And my main thesis is that bacteria are quite clever. They've been around for about 3.5 billion years, and they've been trying to kill each other for about that length of time. So they've had a lot of time to carry out a lot of trial and error. And they've evolved an ability to protect themselves by producing a biofilm, which of course is a kind of gooey, slimy layer of extracellular polymeric materials which protects bacteria from harm effectively. They're like little houses for bacteria. I'm also going to underline the fact that biofilms are held together by DNA. And I'm going to underline a discovery that we made in our laboratory of a marine bacterial enzyme, an endonuclease, which basically chops up DNA and therefore is an efficient anti-biofilm enzyme because DNA is an important part of biofilms. And if you destroy the DNA, the biofilms effectively fall apart. We've commercialized this enzyme into a laundry detergent, which is already on the market, and we're working towards its commercial application in the clinic. And I'll then conclude. So this is an unusual view of the planet Earth, showing from the underside, I suppose. You can just see New Zealand down there on the left, and it underlines the importance of the marine environment. I'm a marine microbiologist, and we've been studying marine bacteria to try and understand biofilms and how they work. And if you look at this diagram, I think we're probably all familiar with the fact that if we look at the formation of the Earth about 4.5 billion years ago, we're probably all familiar with that mankind only appeared a couple of million years ago, right at the in very recent times. And you can see here the red line of the dinosaurs. They were around for a reasonably short time. Mammals, again, fairly short time. But if you look here at this one, you can see that life started about 3.3 billion years ago. So microbes, and including bacteria, have been around for an immense amount of time. And I like to think of bacteria as being, kind of anthropogenic little organisms. They show complex social behavior. They can take decisions on their lifestyles. They can decide to infect someone or not, depending on the situation. And of course, they've been trying to kill each other for many years, and that's led to the evolution of biofilms. And you can see that in the next slide. This is from a paper by an Israeli research group showing really that in a marine situation you can have lots of planktonic bacteria and they can secrete polymers and colloids and these form a blob of gel-like substances called a protobiofilm, which can then attach to a surface and can increase and grow within minutes. And that's exactly the process that occurs on orthopedic joints, and we'll come to that in a moment. But the story that I'm going to tell you today really originates back about in the 1840s, and particularly with the industry of shipbuilding on the River Tyne, and the fact that ships suffered from biofilms growing on their hulls all those years ago. And mankind has been trying to get rid of biofilms from surfaces where they're not supposed to be for many, many years. And our research program began by looking at living creatures in the sea. We were looking at dolphins, jellyfish, seaweeds, and anemones and other things. And we're trying to ask ourselves, why do these organisms remain free from biofilms and slime and fouling? So dolphins were a bit too difficult to catch, so we decided to focus on seaweeds. And to cut a long story short, we were studying bacteria on the seaweeds, and we found that a particular bacterium shown here you can see isolated on agar plates down in that bottom corner, was isolated from a bacteria, sorry, from a seaweed, and they were able to produce chemicals which could degrade biofilms. And we were super excited about this. We knew at the time that we had discovered a chemical which was really, really powerful, and we patented it in 2010, and we identified the active agent, which was dissolving these biofilms, as a DNA degrading nuclease, which was called NucB. And the organism that we isolated it from was a Bacillus licheniformis. So this story begins with seaweed, and we had a very dedicated team at Newcastle University which worked with us, headed up by Rick Lewis's team, to purify and crystallize the structure of this NucB and try to understand why it was so effective at breaking down biofilms. So the answer to that question was the fact that, as I've mentioned in the opening slide, when a cell breaks open, it releases all of its chromosomal DNA. And this is a beautiful slide of an E. coli cell which has been lysed, and you can see all of this spaghetti-like material emerging from the single E. coli cell. And it's an absolutely massive amount of long, gooey, sticky material which exudes from dead cells. And that's incorporated into the biofilm and really acts as an architectural glue to hold the biofilms together. So the key take-home message then is that extracellular DNA outside the cells, is a major component of biofilms. It holds them together and provides them with structural integrity. And therefore you can clearly see that if we then sprinkle on the surface of these biofilms some enzyme which breaks down that DNA, that structural component, then you will get complete removal and degradation of the biofilm. And this is precisely what we saw. Here's an example of NucB-mediated removal of staphylococcal biofilm. Here's an example of removal of yeast biofilm, for example. Again, these biofilms are also held together by extracellular DNA. This is in fact a brewer's yeast biofilm taken from some work that we did with Heineken. And we've also had success in breaking down biofilms of Candida, which of course also has eDNA holding its biofilms together. So we're very excited about the potential of this enzyme to break down biofilms wherever they may occur, of course including orthopedic joint infections. And this slide is really a fantastic slide in the sense that it shows the fact that bacterial cells, and you can probably see a circle forming here. You just see a rod-shaped bacteria kind of starts to blow up and form a circle. And then what you're actually visualizing in this beautiful piece of video footage from the laboratory of Professor Cynthia Whitchurch is the fact that we've got Pseudomonas aeruginosa cells bursting open explosively and releasing extracellular DNA. And the extracellular DNA can be visualized by this fluorescent stain here in real time. So the cells are exploding and they're releasing extracellular DNA. And this happens all the time in biofilms. About 200 events occur each time the cells grow in biofilms. So this is a part of also we're working with Cynthia to look at the importance of extracellular DNA in biofilms, in particular with respect to degradation of that DNA by NucB. We have also been able to work with Dr. Nick Jakubowicz to look at biofilms in oral healthcare. We've seen that NucB also works to prevent biofilms produced by plaque-forming bacteria. And we've also been working with clinical in the ENT department at the Freeman Hospital, looking at bacteria from chronic rhinosinusitis, for example, where we've been able to show that NucB is also able to disperse and break down biofilms of bacteria which are associated with chronic rhinosinusitis. So we think it's only a matter of time before we can develop a clinical application of this enzyme. So of course we've been focusing on orthopedic applications, in addition to some of the other situations we've been looking at with artificial implants, including voice box prostheses. We've been looking at artificial hips and artificial knee joints. And as I'm sure everyone in the audience knows, all of these have serious biofilm related problems. So we've been working on this with some industrial sponsors for a number of years now, and we've been able to pick out some really important key take-home findings, which I'll summarize at the moment. So as you may know, biofilm formation by PJI strains is very variable. So some bacteria produce a lot of biofilm, some bacteria produce very small amounts of biofilm. It's also strain dependent, and it's dependent on growth media. And that's deliberate, okay? That happens because the genetic control of the biofilm lifestyle is deliberately wobbly. And by that I mean that you can have 2 or 3 cells in a colony which start to produce biofilm, and clonal cells, exactly the same genetic makeup and under exactly the same conditions, nanometers away, will not form biofilm. So there's a heterogeneity there, which is completely unpredictable, and that's deliberate. It's a much greater and more flexible lifestyle choice for bacteria. So eDNA is present in the vast majority of single species biofilms that we've looked at, and in virtually all multi-species biofilms that we've looked at. Therefore, it's an excellent general purpose target if we want to go in and attack biofilms by breaking down eDNA. So NucB removes biofilms of clinical strains grown on surgically relevant surfaces. We've looked into, for example, this slide shows titanium, and you can see at the top there blue stained biofilm grown on the surface of titanium, and you can see that when we've treated that with the enzyme, it cleans it very effectively from the surface of those surfaces. We've also done the same for high density polyethylene with and without NucB, and we've seen that it's very effective at breaking down biofilms. This slide also shows the effect of NucB on Staphylococcus epidermidis. Again, it's a clinical strain, and this prevents biofilm formation. Okay, so not only does it break down biofilms, but it actually stops them occurring in the first place. If we look at the next slide, we can see that we've done the same experiments with Staphylococcus aureus, and we found that it stops them dispersing as well. So NucB causes biofilm dispersal of established biofilms, and it also stops them forming. So I think we've basically generated some foundational data and evidence that suggests that we can commercialize this enzyme into some kind of a therapeutic substance which can be used to prevent or stop or remove biofilms from particular surfaces. But how do you then go about commercializing such an enzyme? And that's the subject I'd like to talk about in the next slide. And it's a slight shift in thinking, particularly for this audience. I'd like you all to suddenly start to think, if I may draw your attention to dirty laundry. So we've been working with Procter & Gamble, which is a manufacturer of laundry detergents, amongst other things. And interestingly, Procter & Gamble was one of the first companies that approached us back in 2010-11 when we spoke to a number of companies and said we've got a biofilm destroying technology here. They contacted us and started working with really early on. So they were very early adopters of this new technology, and they were kind of willing to take risks on the technology, which was very much unproven back in those days. And the collaboration basically led to a very successful commercialization of NucB-based enzymes. And why is this relevant to a conference on infection in orthopedic infection? I think that can be clarified by the next slide. And this is a paper from a German group which looked interestingly at community analysis of all of the microbes in different washing machines containing different types of laundry. So they did a very forensic approach, and the conclusions were not really that surprising. If you can imagine that your clothes are probably contaminated by bacteria such as Staphylococcus aureus, Staphylococcus epidermidis, and Pseudomonas aeruginosa, you wouldn't be surprised then to find that these are the most common microorganisms which establish themselves on laundry and which need to be cleaned. And we then worked with Procter & Gamble to study laundry, and effectively found that if you look at what might ostensibly be a clean t-shirt under the naked eye, then something that you often cannot see are molecules which are left behind on the fabric, such as DNA. And you can find DNA on even clean t-shirts, and it's because it's not cleaned by traditional laundry detergents. It sticks to the fabric, and the origin of that DNA is found to be associated with microbes which grow on the clothes, and of course which come from the skin of the wearer of the t-shirts. And so what we can see then is that these molecules of DNA which are on the t-shirt have an interesting function, and they serve effectively to stick dirt onto the clothes. Okay, so even if the t-shirt looks clean, it's covered in this layer, if you like, of bacterial adhesive molecules, including extracellular DNA. And what we noticed when we worked with Procter & Gamble is that the addition of NucB nucleases caused the removal of the extracellular DNA on the laundry. And that in itself had a really interesting effect. It caused the laundry to be have the eDNA removed, and that stopped the dirt from sticking to the t-shirt, so the t-shirts came out looking cleaner. Because at the molecular level, the fabric of the t-shirt was less sticky, also microbes stuck less to the laundry, and in fact, when the laundry was put out to dry, then there were less sort of malodor associated with laundry, and also, interestingly, things like the ironing, the ease of ironing improved as well, because these molecules were being stripped off the t-shirts. So, the great news about this was that this represents a new class of enzymes to be formulated into laundry detergents, and they were launched. This was launched just last year in 2020, and in fact, NucB-based nucleases are currently found in all aerial products. If you go out to Tesco's today and find yourself some aerial pods, then the Newcastle-based nuclease enzyme has been commercialized into those laundry detergents now, and it was amazingly advertised at the Great British Bake Off launch last year, which had 9 million viewers, and it appears to be growing into a tremendously successful application of a nuclease to break down and take away extracellular nucleases where we don't want them. And we believe that this demonstrates the power of this approach, and we believe that although the commercialization of the enzyme into a biomedical product which will be much more difficult, we believe it's only a matter of perhaps a few years away. So just to close, we patented this in 2011. A US patent was granted in May 2019, and we commercialized the enzyme in 2020. And we're currently working with an industrial partner to look at the commercial application of this enzyme in biomedicine. So to conclude, we've developed an enzyme-based novel biofilm destroying technology. The protein is stable and highly adapted to dispersing biofilms. We've commercially applied it to laundry detergents, and we're looking to commercialize that into a medical device. I'd also like to thank the team, which has been led by Professor Mike Reed, and also colleagues Mike Hall and Anjam Khan at Newcastle University, as well as a stream of medical doctorate students and the Newcastle Proteomics facility as well. And with that, I'd like to thank you. This was a diagram I prepared many years before we've commercialized it, and we started from seaweed. We isolated the bacterium and the protein. We've applied it to breaking down of extracellular DNA, and we've been really fortunate in being able to apply that to products which are on the shelf today in a little under 10 years, which is relatively fast from discovery to commercial application. And we'd like to repeat that success in the therapeutic medical device area in the coming months and years. Thanks very much for your attention. And again, my apologies for not being able to be there in person, but do please drop me an email and I'll be happy to discuss this project with you as well. Thank you again.